All right, everyone. We're going to go ahead and get started. Thank you, everybody, for being here at our seminar. My name is Cinnamon Moffat. I'm the Associate Director of Marine Research Support here at Hatfield. I'll be your host for today. This is a hybrid event, so we have folks online and folks in the room. So for folks online, thanks so much for being here. If you have any issues, any questions, go ahead and use the chat function. We have a volunteer, Roseanne, in the room who will help navigate those questions, and we'll read them out to today's presenter. For all of you here in the room, if you haven't been with me before, I'm looking around, I see a couple new faces. When we get to the question and answer section, all you need to do is raise your hand and I'll bring you this mic, and that way folks online can hear those questions as well. So we'll navigate it that way. And we have some show and tells coming into the room, which is awesome. Okay, I wanted to do a couple of quick little promos, and then I'll get us rolling here. So next week's seminar will be Nick Sentner, who is from Central Washington University. He will be talking about how did lava flows from eastern Washington end up in Uquinna Head. So that one is going to be a fun one to come and learn a little bit more about. I also wanted to promote next month's Science on Tap. If you haven't heard yet, we're going into a shark month. There's going to be all sorts of shark things happening next month. So our next Science on Tap will be Wednesday, March 18th, and we have Isla Hoganson, who will be here talking about how humans interact with shark conservation. So hopefully you can join us on Wednesday the 18th, and that'll be an evening event starting at 6. But if you need any information about any of our talks, anything that we do here, if you go to the Hatfield website and you go to the events page and you scroll down, you'll see a calendar there of everything that you might need to join us either online or in person. We try to hold these events both hybrids, so you can join us however feels comfortable for you. But for today, we're very excited to welcome today's speaker, Kathleen Hunt. Kathleen studies stress and thyroid function and reproductive in a wide variety of vertebrates, often using alternative sampling types like feces and vapors and hair and baleen to figure out hormone patterns over time. Her research focuses on sublethal effects of repeated and chronic stress on health, reproduction, and metabolic rates of wildlife. And she has spent 25 years developing these novel, but these novel essays to about dozens of mammals, birds, and reptiles. And this list was pretty impressive, so I'll just kind of give a couple, looking at pinnipeds and large whales, but also elk, moose, wolf, lions, elephants, bears, oh my. So that is pretty crazy and cool. And so she did her PhD in the University of Washington and has worked both for conservation and academic organizations before joining us here at Hatfield and MMI as the newest associate professor. So we're very excited to have you share with us. Turn on your milk and welcome.
Thank you so much, Cinnamon. All right, let's get going. So I'm really excited to be here to talk with you today. I have a number of different projects I've been doing to look at these hormones in a variety of marine mammals, but I mostly am working on large whales right now. I have decided that we are going to zero in today on whale baleen, which has become one of my all-time favorite sample types. So let me first back up, though. I just realized I don't know how to go forward. Wait a second here. Oh, okay. I'm going to back up and give you sort of an overview, though, of what I'm actually trying to learn. Why am I using hormones at all? So I'm a physiologist. So what is physiology? Physiology is really the study of how the body works. So physiologists look at individual animals, and we're often trying to peer inside their bodies and see what is the heart doing, what is the liver doing, what is the reproductive system doing. Anatomy is what it looks like. Physiology is how it works. So that's generally physiology. And so as a physiologist, I really am interested in individual animals. And more specifically than that, or maybe more broadly than that, I'm an ecophysiologist, which is really the study of how the animal's body responds to the environment, to the ecosystem around it. If something's going wrong in your environment, what does your heart do? What does your adrenal gland do? What does your reproductive system do? I'm really interested in stress and reproduction most of all. So I spend a lot of time thinking about and trying to study how individual animals are affected by stressful things that are happening around them in the environment. Some of those are natural things. Some of them are caused by humans. And then stress often interferes with reproduction, sometimes immediately, sometimes down the road, a year or two. And ultimately, you'll end up with population changes, with changes in reproduction and health and mortality resulting in changes in population numbers. I have here bears as my example because, as you just heard, I wasn't originally a marine mammal biologist. I started working actually on small birds and then spent a lot of time on the large terrestrial mammals. I did a lot of bear and elephant studies, so I really kind of cut my teeth on the stress and reproductive hormones with terrestrial animals. And as you will see shortly, there's a lot of challenges when you try to move those techniques to the marine mammals, and that's been really interesting. When I'm studying stress and reproduction, I'm often trying to study the hormones that are involved in stress and reproduction. And I want to just give you a quick introduction to the three groups of hormones that I'll be studying today, whose names are probably somewhat familiar to you. So first off, you'll see immediately these all look really similar. These are the steroid hormones. They all have this classic shape where they've got these 4 interlinked rings. But if you look really closely around the edges, you'll spot some little differences between these hormones. So what I'm going to be showing you data of today is going to be from data on progesterone, which is the great pregnancy hormone of the mammals. It's high in pregnant females. It helps keep the pregnancy going. Testosterone, most famous for its role in males, so it's going to be high in breeding males and sexually mature males during breeding season. I will say I can't skate over testosterone without mentioning females also have and use testosterone, and males also have and use progesterone, so they're actually present in both sexes. But I'm going to be focusing on what testosterone's doing in the males. And then there's the stress hormones. There's actually multiple other types of stress hormones than the 2 I'm showing here, but I'm going to be focusing on these 2, which are 2 from your adrenal gland. And we and the whales have the same hormones, so the whales have the exact same hormones as shown here, and so do humans. You might have heard of cortisol. It's gotten rather famous as the hormone that goes up when you're stressed. But there's another very similar one called corticosterone, and we actually have both. We just have a little more cortisol. Whales have both as well. I'm going to be showing you mostly corticosterone data. But think of those as the stress hormones, those glucocorticoids. So I would like to be able to measure progesterone and testosterone and both those glucocorticoids to see what's going on with stress and reproduction inside an animal's body. It would be really helpful if I could get blood samples. So I was trained on, as I said, small birds and bears, and actually I took that photo on the left. That was a bird project I was involved in. So I was accustomed to being able to hold the animal in my hand and get a blood sample from it. I can't do that with the whales. And even with the bigger animals like bears, we usually had some way we could get hold of the animal, even if we had to tranquilize them, and we could get blood samples. And classically, the most traditional way to measure hormones is to measure their concentration in the blood plasma. And so I was trained expecting that I would always be able to get blood samples from animals to measure these stress and reproductive hormones. Then I started working on the whales. And so there's multiple issues when you try to do this sort of research in whales. One big one for me is we can't get blood samples. We sometimes, not only can we not get blood samples, we can't catch them at all, these large whales. Not only can we not catch them, we often can't even get close enough to get a good picture. There's the view we want, and then there's the view we get. And it really kind of blew my mind as a bird and bear trained biologist who was out on the boats for the first time. It really blew my mind to realize I was now working with animals where I could never catch them, never get a blood sample, never weigh them. And at the same time, these were also animals where there were other huge data gaps, things we didn't know. We didn't know how often they bred. We often don't really know how long gestation is. We have a guess, but it's really just an estimate for some species. Sometimes we don't know where they go for part of the year. We're not sure when breeding season is. So these are what we call data gaps, things we don't know. Huge data gaps, no possibility of blood samples. So that's problem number one, is I can't get blood from the whales to measure the hormones. There's a couple other problems too. The things I'm interested in play out over time. You get stressed one year, you don't breed the next year, you don't have your cub or your calf the year after that. 10 years down the road, you have the population impacts. This is multiple years of effects playing out over time. And really to study this well, we would want repeated samples over years from the same individuals. So that's problem number two. How do we get repeated samples over time so that we can look at these long-term effects of stress? Then as I started grappling with ways to deal with problems 1 and 2, I ran into problem number 3, which is that we're now developing techniques to get around problems 1 and 2. I can now go out and measure progesterone from some other sample types from whales that I'll tell you about in a second. Suppose I grab some other sample type, a fecal sample or whatever it is, and I run it through my hormone assay, and I get a result. Progesterone is 15 nanograms per gram of sample. Is that high or low? Is that normal or not normal? The problem I'm facing now, problem number 3, is that the world has changed dramatically in the last couple decades and is continuing to change. And almost always, when I go out and study a wild population today, I'm dealing with a population that is, to some degree, trying to cope with a rapidly changing environment, and they may not be that normal. They may not be breeding normally. Their stress hormones may not be normal, but I don't know what normal is. I don't have a normal to compare to, because to get the normal, I would have needed to have already been studying these hormones 30 years ago and measured normal back then and be able to compare to that baseline. So this is the problem I think that all biologists today are grappling with, that the populations they're studying are not at normal anymore, and we don't have a good comparison point anymore for what to expect a normal, unstressed, healthy individual to look like. So problems 1, 2, and 3, bear those in mind as I start playing out this story about struggling to look at hormones in different types of things. So let me first address the problem that we can't get blood. It turns out that hormones are present, I now believe, in every single tissue type in your entire body, especially those steroid hormones that I showed you before, progesterone, testo, and the 2 glucocorticoids. Those are small molecules. They're non-polar. For any of you struggling to remember that term from way back in your high school chemistry days, that means they can move through fat tissue, they can move through cell layers, they can get through tissues in the body really, really well. And it turns out the steroid hormones sort of permeate the body. They're present in the poop. That was the first unusual sample type I was working with. They're present in respiratory vapor, which is the next one I started to work with, the vapor that a whale blows out when it exhales. There's hormones in that respiratory vapor. And I have colleagues who are working on whiskers, or they're formerly called vibrissae, of the pinnipeds. I have another colleague working on bone hormones from walrus bone. I have a whole bunch of colleagues who get little biopsy darts of skin and blubber, and we've discovered there's hormones in the blubber, and I now know there's also even hormones in the little bit of skin on top of the blubber. And then there's baleen. We only get baleen if a whale dies. We can't get it from a living animal, but unfortunately whales do die pretty often, and it turns out there's a lot of baleen that's been collected over the years in the stranding archives and in the natural history museums. And it turns out, a bit of a spoiler, but there's hormones in baleen as well. So if we can get any of these other sample types, any other piece of a whale or any excretion from a whale or anything that's coming out of the whale, we'll be able to measure the hormones in that sample type. So that's been really useful. I call these the alternative sample types, really meaning they're not blood, and we don't necessarily need to catch the animal to get the sample. However, you then need to think really carefully about what that number means if you measure the concentration of a hormone in a poop from a gray whale or in a bit of blubber from an animal that you've darted. When was that hormone put into that tissue, and what time frame are we looking at? I just brought up a whole bunch of other bits of text on this slide. For each sample, I'm showing you roughly what we think is the case for if we measure hormones in that sample, what time frame is that telling about in the animal's life? For example, respiratory vapor, we think the hormones in the respiratory vapor were probably in the blood 30 seconds ago, like just before the whale exhaled. So we're getting a little snapshot of the whale's hormones right in that minute that it was exhaling. Feces, we now know, is actually telling us about the whale's hormones the day before, because it takes feces about a day to work its way through the gut and come out the other end. And it turns out the hormones that are in a fecal sample are actually the hormones that were circulating in the blood yesterday. So you can look back in time 1 day with a fecal sample, 1 to 2 days. And so the time frames vary with all these other sample types. And I think you're seeing already with the baleen at the bottom, you've got multiple years of information in the baleen, and that's what I'm going to be zeroing in on. So baleen is long and skinny, and I'm going to spend actually quite a bit of time describing baleen to you because it's bizarre and really cool and has been really useful for measuring hormones. But first, let me show you some similar structures from other animals. Baleen is made of keratin, and let's take a look at a couple other keratin tissues. Keratin is the protein that your hair and your fingernails are made of, and it's a really sturdy protein. A lot of mammals, in fact a lot of vertebrates, have keratin somewhere on the outside of their bodies. It's nice and waterproof, it's very sturdy, it makes a good outer coating. And it turns out if you have some evolutionary need to evolve a bristle of tough hairs on your tail or a horn on the front of your snout if you're a rhinoceros, you end up often making those structures out of keratin because it's so sturdy and stable. So there's a lot of mammals that have evolved some kind of long, skinny thing that's growing out of their skin that's made of keratin. And some of these tissues grow, I call them the linearly growing samples, where they're growing continuously from a base region, and they're extending outwards as they grow. For example, all the 4 photos on the right there, we've got long tail hairs of an elephant, we've got a turtle claw, we've got a rhino horn, and we've got a sheep horn. All of those are linearly growing samples that are growing from a base region, and it turns out they grow slowly over time, and they are incorporating hormones as they grow, such that the further away part of the sample has hormones from several years ago. And the part that's just growing right now is gathering the hormones of today. And if you take a bunch of little samples along the length of that sample, like shown by the yellow dots, if I take that rhino horn and drill a little bit of powder on each of those yellow dots, I've got a time series. I've got a bunch of samples that were laid down at different points in time from that animal's life. So some samples have this feature of having this time series. Some sample types don't. I've got a couple other sample types on the left that don't have a time series. So it turns out, we now know after years of effort, it turns out maned wolf hair is not a time series. If you can measure hormones in maned wolf hair, but it just tells you about 1 month when all that hair was growing. It doesn't go further back in time than that. Respiratory vapor from a whale is telling you just about that 1 minute, so that's not a time series. I have a snake skin showing in the lower left. We've got hormones out of snake skin too, but it turns out the snakes grow that whole skin all in the same month. So it tells you about that month, but not further back in time than that. So I ended up getting really interested in these time series samples because they solve that problem of getting repeated samples over time, right? So that brings me back to baleen. Baleen is a time series keratin tissue. So I'm going to spend quite a few minutes here just telling you about how baleen grows, because it's really interesting. It takes a little bit to wrap your head around if you've never stared much at a whale's mouth before. So this photo shows a Brutus whale, which is one of the baleen-bearing whales, or Mysticete whales. He's in the middle of gulp feeding. I chose this photo because you can see, with his mouth open, his lower jaw is facing us, and his upper jaw is kind of up there in the air. And I think you can see that upper jaw's got 2 big fringes, one on the right side, one on the left side. It looks kind of short. It's actually about 1 meter long. This whale is really big, even for, I know Brutus whales aren't that big for Mysticetes. They're still a big animal. That baleen's actually pretty long. That fringe of white on the 2 sides of its upper jaw there, each of those long fringes is actually a whole series of 100 or so, actually several hundred, little pieces that we call plates. So in the upper right there, you'll see a close-up view. That's actually a Rice's whale, where you can see, again, that fringe of baleen. Again, this is strips of keratin. And I think you can see there it's anchored in the upper jaw. It's growing from the upper palate, growing continuously from a well-vascularized area in the upper palate. And then the lower tip is free-hanging. The lower tip is not attached to the lower jaw, right? And if I take just one little piece of baleen out of the side of that whale's mouth and zoom in on it, that you can see in the lower right there, that piece we call a baleen plate. And that was grown over some span of time. That particular plate is actually from a fin whale. So it's about, for fin whales, that's going to be about 5 years of growth. So I can take little bits of powder, add all those little yellow dots all the way down the animal, and I've got a time series. Some animals have much longer baleen. Some whale species have longer baleen than others. The bowheads and the right whales have the longest baleen. This is a skeleton of a southern right whale on display at a museum in France. I really like this display because not only did they mount the skeleton, they mounted the baleen too. So you can see, I think, how this right whale has dramatically long baleen. And we're getting a really good view of the right side of its mouth there. So again, it's anchored in the upper jaw. We call that the base because that's where it's anchored, that's where it's rooted, that's where it's growing from. So the base is on top. I hope that's clear. And then as it's growing, it's slowly extending downward, and then the tip is getting eroded away as the whale feeds. So just to sum up what we learned about baleen, baleen is present in what we call the baleen whales, or the Mysticete whales. So if you think of all the really large whales, there's one with teeth, right? A sperm whale. That one's not a baleen whale. All the others do not have teeth, and they have baleen instead, and those are the Mysticete whales. So blue whales, fin whales, gray whales, humpback whales, bowheads, rights, a couple others. Those are the Mysticete whales. This is their filter feeding apparatus. It's an amazing evolutionary innovation, and what they are doing is taking in water and their food into their mouth, and then basically the water gets sort of flows out through the little slats of baleen, and the food gets stuck on the inside, and then they scrape it off with their tongue and swallow it, and that's how they filter feed. So again, it's made of vertical strips of keratin. I'm going to now zoom in again. I just explained baleen to you, but I'm going to explain it again because I want you to get the idea of the shape of one long piece of baleen in a right whale. So right whales have particularly long baleen, and I've sort of outlined in blue one piece or one plate of baleen for a right whale. Again, it's growing from the base, it's extending downward, the lower tip is free hanging, not attached to the lower jaw, it's free hanging. It's just leaning on the lower jaw in this particular mount. And again, I can take a bunch of samples from along the length of the baleen. So when I'm going to be showing you a bunch of graphs of baleen plates with this little blue triangle kind of indicating where the baleen is and where I'm sampling along the baleen. And I want you to remember that the tip is the oldest baleen. It was grown several years ago. And whoops, sorry, I went ahead and I meant to go that way. The base, the widest part of this slat, is the newest baleen that was grown typically like the day before the whale died is when that last little bit of baleen was grown. So we've got the base with the newest baleen, the tip with the oldest baleen. So that's baleen. Now we've known for centuries that these large whales have this thing called baleen. And it's been known that baleen grows slowly over time. And there's a whole field involving analysis of stable isotopes in baleen. There's a whole field where people have been drilling little bits of baleen powder along the length of the baleen and analyzing it for ratios of stable isotopes, which I'll tell you about in a second. So it's not a new thing to know that baleen grows slowly over time. The new thing is realizing that it's got hormones too. Now the way this came about is, as I've told you, I was working with bears and elephants, and we do, though we can get blood from bears and elephants, often that's rather difficult to do. And so we were kind of rummaging around looking for other sample types we could study in bears and elephants, and we started using fecal hormones, so I spent a long time working with poop of bears and elephants. And then we started working with hair, grizzly bear hair. Did a long project on grizzly bear hair. And I realized that there were a number of other teams that had discovered that you can measure hormones in mammal hair. And then, if you look at baleen, that is a photograph of bowhead baleen there. Look at that little fringe in the lower edge of the baleen. And those of you who are online, I don't know if you can see me clearly. I have here a piece of right whale baleen. See this fringe? And I want you guys all to come up. Feel free to come up after the lecture and take a look at this. This is hair. If you look in the textbooks, it'll say, there's a fringe of remarkably hair-like material on the inner edge of the baleen. Remarkably hair-like. This is strands of mammalian alpha keratin. This is hair. This is mammal hair. I'm convinced. So I was working with grizzly bear hair. There's this hair-like fringe on baleen. And the natural next step was to think, could there be hormones in baleen? Though actually, I was not the one who had this idea, even though I was working on grizzly bear hair. It was actually a colleague of mine at the North Slope, a wonderful wildlife veterinarian by the name of Rafaela Stimmelmeyer. She's the one who put two and two together and emailed me and my colleague and said, I see that there's hormones in hair. I'm looking at a piece of bowhead baleen. Looks like it has hair coming out the side. Could there be hormones in baleen? And I immediately thought, oh my God, why didn't I think of this? So I did not have the idea. But fortunately for me, Rafaela wanted to work with me and my colleague. So we thought, well, let's just give this a try. And I read all the hair hormone literature I could find. And there's also, it turns out, a whole feather hormone literature as well. So there's also hormones in bird feathers, which is also made of keratin. And I ended up just trying sort of the average method that people in those fields have been doing. And long story short, we spent 6 months trying to devise better, cleverer ways of pulverizing baleen. And we ended up just getting a $65 Dremel from Home Depot and just drilling a little groove in the baleen, and it made this little powder, and that was perfect. So you just need to get a little bit of powdered baleen, and you stir it, and you weigh it, and you then mix it with, it turns out, any alcohol will work pretty well. Steroid hormones love alcohols. They love to move into alcohols. So you just mix the powder with an alcohol. I just use methanol, and the hormones will move right into the methanol. And now you've got it out of the powder, and now you can now you've got it in liquid form. From there on, you can use pretty standard lab techniques to measure it. I happen to use one called enzyme immunoassay, often called EIA or ELISA for short. I won't tell you anything more about this method right now, but in case any of you are interested, I'm in the middle of setting up a lab here, so we'll be able to run ELISAs here for all the hormone projects I am running and any other projects you guys might have that might need ELISAs. So it's a tried and true method of just measuring concentrations of, tiny concentrations of things that are in liquids. So we just did that with some bowhead baleen, and just our first try was just to see, well, let's look for progesterone. Is progesterone even there? Immediately, yes, it was there. There was a lot of it. It was easily detectable, and instantly we had just launched into what I call the validation phase of starting a new sample. Whenever you devise a new technique, you end up having to do a bunch of validations, and there's many, many types of validations, and validations can take your whole life if you're not careful. But long story short, in the hormone world, we tend to think of this in two subphases. One is, can our assay put a number on the concentration of the thing we're trying to measure? Can it actually detect that thing, that molecule? The answer to that was pretty clearly yes, and there's a bunch of tests I did to verify that I'm just going to skip over right now. But the assay validations were looking really good. So now you've got a number. Now you've measured your progesterone, and it's, say, 128 nanograms per gram or whatever it is. Now you have this other type of validations you need to know. So it's 128. What does that mean? Does that tell you anything real about the animal? Does it really correlate with whether or not that was a pregnant female? Does it tell you anything useful? Does it match the biological reality of what was going on with that animal? So I think of this set of biological validations as being about, do the hormone data tell the truth about what was really happening to the animal in its physiological state? So it turned out when we did our first little try at baleen hormones, we happened to have some very recently grown baleen from a couple of pregnant females. And we had some very recently grown baleen from a couple of non-pregnant females and from some males. And I'm showing you the graph there on the right, and I think you can immediately see progesterone was much higher in the pregnant female. So right away, we had a big clue that the biological validations were going to work out, we think. But this was a small sample size. Our extraction method actually wasn't that well worked out yet, but also this was only the most recently grown baleen. It was just that root region, that base region right inside the upper palate that had been right next to the circulating blood. We still weren't sure if the hormones were really going to be in the further down baleen. And what I really was unsure about is would hormones last in the baleen? Because the baleen grows slowly over years. Would the hormones still be there 5 years later in the older baleen, or would it have all fallen apart? So we still needed to do a whole bunch of biological validations in animals where we knew when they'd been pregnant in the last several years. We needed animals of known state. And specifically, I realized we needed a female whale with known calving history, where we already knew that individual female. We already knew when she'd had babies. So, and then we needed to figure out the timeline. How fast does baleen grow in the mouth of a North Atlantic right whale or a bowhead whale? And this is where I immediately started leaning on my stable isotope friends. So more about that in a second. So we need a whale of known calving history, and we need to figure out the timeline before we're going to be able to see, can we really use baleen to sort of look back in time to the prior years? So a little bit about the timeline. We're really fortunate with these Mysticete whales. They migrate. They're famous for migrating. I have here a map. This happens to be a map of humpback whale migrations. It's just an example of migration routes of a baleen whale species. But notice how long these migrations are. These Mysticete whales often have very long migrations where they're going very far toward the poles in the summers to productive summer feeding grounds. And in the winters, they come down closer to the equator, usually to calving grounds, and sometimes their mating grounds are right by the calving grounds as well. They switch what they're eating in summer and winter. There's different prey in those two locations. Some species stop eating and just live off their body fat. That actually counts as a prey switch. They're now eating themselves, so they're eating their own body fat, and that actually molecularly that translates to a kind of a switch in prey. And it turns out different prey or your own body fat have characteristic ratios of carbon 13 to carbon 12, and nitrogen 15 to nitrogen 14. So these are chemical elements, right? Carbon and nitrogen. And you may remember that that a chemical element has a certain number of protons and neutrons in its nucleus, and every now and then there's an extra neutron, and that's a new isotope. So most carbon has 6 protons, 6 neutrons, carbon 12. And every now and then there's a carbon atom that has an extra neutron, carbon 13. It's rare, but it's out there at a certain characteristic amount. And same with nitrogen 15 and nitrogen 14. So you can look at the ratio of the rare isotope to the common isotope for both of these elements, carbon and nitrogen. And you can look at that ratio in different prey species, and it's different in different prey species. So You can take baleen from, in this example, a bowhead whale, and get baleen powder from different points all the way down the baleen. So this is a real graph of data from a real bowhead whale's baleen. And you can look at the ratio. In this case, it's the ratio of nitrogen, the rare nitrogen isotope to the common nitrogen isotope. And as you look along the baleen, you'll see that ratio going up and down, up and down, up and down. See that? Up and down. The ups are summer prey, and the down is winter prey. Summer prey, winter prey, summer prey, winter prey. We're seeing its annual migrations. That tells us how long a year is on the baleen plate, and it tells us the baleen growth rate. So you often will get these lovely annual cycles of stable isotopes, and that gives you your timeline. And I'll tell you, for example, this girl here, this baleen plate from a lovely lady named Stumpy, a North Atlantic right whale, she's got 9.5 isotope cycles along the length of this piece of baleen. So we knew that this took 9.5 years to grow. More on Stumpy in a second. She's going to be my poster child. So we now know how to figure out the timeline. And from my point of view, what I learned is I need to go find some stable isotope friends who already know all about stable isotopes. So I do a lot of collaborations with stable isotope people who have already worked this out for many populations of whales. Stable isotope people are usually most interested in what whales are eating. They're looking at feeding ecology. And they just sort of, as a byproduct, end up figuring out the timeline of the baleen. And then I came along and I was like, give me your timeline, please. I need to know which year each of these areas of baleen was grown in so that I can make sense of my hormone data. So we got the timeline now, or at least we know what to do for the timeline. Now we need a whale of known calving history. And I will never forget this particular piece of baleen. I was working at the New England Aquarium at the time, and I had this moment of like, I need a whale of known calving history. And I walked into my boss's office, Scott Krause, who was director of research at the New England Aquarium at the time, and said to him, Scott, if only I had a baleen plate from a female where we knew when she'd be seen with a calf in like every year of the last 10 years, and we'd like surveyed the calving grounds every year for 10 years, and we knew when she'd had a calf. And Scott literally moved past me, reached behind his door, pulled this out. He's like, here, Stumpy. And she was a known adult female right whale, and we'd surveyed the calving grounds every year for 10 years, and we knew when she'd shown up with babies. And Scott had a piece of her baleen behind his door. I still can't get over this. So this is Stumpy, who actually is a real tragedy. So she's a North Atlantic right whale, and they are critically endangered. And every North Atlantic right whale lost is a tragedy for the population, but especially an adult breeding female, which she was, especially a pregnant adult breeding female, which she was. She died, she was first studied in 1975. She died in 2004, February 2004. She was hit by a ship. And this is Stumpy's carcass here. And this, the fellow in the high-vis jacket there, by the way, is Michael Moore, a veterinarian and large whale specialist who works at Woods Hole. He attended her necropsy. He took it upon himself to try to save some of her baleen, even though at the time we didn't know if it was ever going to be useful for anything, and nobody was thinking about hormones in baleen at that point in 2004. But he took some baleen, which is really difficult to do, and got it back to Woods Hole and stored it in case it might be useful someday. And I'm forever grateful to him for doing the effort for saving those samples. Side note here, I'm really grateful for the stranding response teams all around both coasts of the USA, because they've been incredibly helpful colleagues. Stumpy was killed by a ship on her way to the calving grounds, and she was almost there. She was probably just a couple days out from getting to the calving grounds, and she was pregnant with a full-term fetus. She was probably just within a week or two of giving birth. It was a tragedy. But now at least we have the opportunity to learn something about Stumpy and learn something about North Atlantic right whales and other right whales. All my baleen comes from an animal that's died. I feel this responsibility to learn anything we can from those cases. So let me walk you through the timeline again. Part of why I'm telling you so much about the timeline is I've just started here at the Marine Mammal Institute. I want to collaborate with a lot of people in this room on baleen hormones, and so I'm basically training you all right now, and everybody online, all of you, whether or not you work at the Marine Mammal Institute, you are all my potential collaborators, and I'm training you up so that we can work together. So let's go through working out the timeline for Stumpy. So there's the baleen plate. So this, again, those of you online, I hope you can see this. This is Stumpy's baleen right here. I'm holding a piece of Stumpy's baleen. So this is the base, most recently grown. This was grown February 2004, the week before she was killed. This is the tip. It was grown some time ago. How do we know that timeline? How do we know how many years ago? Because of the stable isotopes. So let me walk you through what we know before having to turn to isotope data. We know how long this is. We can measure it. It's about 220 centimeters long. We know that time is going, this is back in time at the tip, and we're going forward in time as we move to the base, right? And the most recent time point is over here. We tend to measure the base as being 0. We call that the 0 centimeter point, and then everything else is measured as centimeters backward from the base. But time is going this direction, from the tip toward the base. We know the day she died, or rather, we know the day she was found floating dead, but she was pretty freshly killed, so we think she was killed either that day or the day before. So we got a pretty good fix on the day she died, and that's the date of the last date of growth at the very, very base of the plate, right? What we don't know is the date at the tip of the plate and how many years there are. So this is where we turn to the isotope cycles, and as I mentioned before, I gave you the spoiler already, but if we count up her isotope cycles, there were 9.5 nice isotope cycles, and we were able to figure out her baleen growth rate. And I have to give a shout out to my colleague Nadine Lysiak, who was the person who had all the isotope data. Scott just had this baleen plate behind his door, and then I was saying to another colleague, I wish I knew someone who'd done isotopes on right whales, and Nadine's like, I'm doing my PhD on North Atlantic right whale isotopes right now, and I just ran Stumpy's isotopes. So it just kind of magically all came together. So from the isotope data, we knew this was a 9.5-year growth sequence, the 9.5-year time series. If you then do the math and calculate the estimated date of growth along her plate, this is what you get. It starts in late 1994 and goes forward about a decade and ends in February 2004. So now we go to the sightings record, and this is where I called up Philip Hamilton, who at the time was running the North Atlantic right whale photographic ID catalog. And I said, Phil, what do we know about Stumpy? And he sent me all her sightings records. And I was able to divide them up basically in terms of was she with a calf or not. And it turned out, even though we do intensive surveys of the calving grounds for the North Atlantic rights every year, she had only been seen on those calving grounds one year before, and it was late 96 and 97. She was seen with a very young neonatal calf at the very end of 1996. I've shown that with a green dot. That's a brand new baby right whale calf. And then she was seen with that calf over about the next 10 months as she was nursing that baby, and that's about the normal lactation duration for a right whale. The black dots are when she was seen in other places with no calf, and she was never seen at the calving grounds in those other years. And then there's the green triangle at the far end. That's that full-term fetus that we found when we did the necropsy. So she only had 2, for sure, pregnancies. And there's a lot of years in between those 2 calves, and one thing we were wondering is whether she might have had an undetected pregnancy where we just never saw the baby. Somewhere in the middle. Right whales are, in theory, supposed to breed every 3 years, and she should have had another baby like around the year 2000, and we didn't see her with one, but I was kind of wondering maybe she'd had another pregnancy in there that we just hadn't observed. But we did have 2 pregnancies for sure. So she had to have been pregnant the year before those 2 babies, right? Because here's where I have to put a little caveat in. At the time, we believed that gestation length for North Atlantic rights was about 12 to 13 months. And you'll see in just a moment why I'm putting so many caveats around that. But 12 to 13 months is what was in the textbooks and is still in the textbooks right now, and I'm pretty sure it's not correct. But anyway, the yellow bars show 13 months before the arrival of that neonatal calf and 13 months before that full-term fetus. So she should have been pregnant during those yellow bars. So I'll take that baleen plate triangle away, and I'm about to finally show you the graph. I sampled her baleen plate all the way down, and then I assayed for progesterone, and I'm expecting higher progesterone in the yellow regions and lower progesterone elsewhere, and here's what we got. And it is to this day the prettiest data set I've ever generated, and it was actually really jaw-dropping because look at the y-axis. This is a log scale. Her progesterone when she was pregnant was 100 times higher, orders of magnitude higher during pregnancy than during non-pregnancy. And by the way, notice there's no indication of a pregnancy that we didn't observe in the middle. It looks like she just didn't get pregnant for 6 years in a row right there in the middle. So this was stunning, and this was the strongest confirmation I could have wished for that yes, the steroids are still present even in old baleen. And at this point, not only did the baleen take 9.5 years to grow, but also the baleen plate had been sitting around at Scott's office at room temperature for another 10 years. So this was like 20-year-old hormone molecules in the end that I was looking at, but they were still there, still detectable, still looked pretty much like the more recent pregnancy. And then, so I really had done this as a validation to demonstrate whether or not baleen hormones tell the truth about the animal's past. This is just one individual. I was able to then get another female, also of known calving history. Same story with her. The baleen progesterone patterns perfectly matched her calving history. We then got other individuals, and we got some males, and we got some animals that had known entanglements, and we did many other studies. And long story short, without showing you a billion other papers, we think that baleen hormones are telling the truth about the animal. So this was conceived of, and I still describe it as a validation study, but we immediately learned some additional things that we didn't expect. One thing was the fact that she totally skipped a calving cycle that she should have had around 99, 2000. So I don't have much time to tell you too much more about this, but I will just mention that a lot of North Atlantic right whale females stopped calving around 1998, 1999, 2000. There was a crash in calf numbers, which is actually why the New England Aquarium team reached out to me to see if I might be interested in working with them. They were worried about the crash in calving numbers, and that's how I ended up moving into marine mammal biology. We think now that there was a disease coming through the population at that time, because a lot of whales ended up with these kind of odd-looking white lesions on their skin in the late 90s, and a lot of females just seemed to completely stop ovulating at that time. So what I'm reading this as Stumpy just didn't ovulate for 6 years in a row. We still don't know what that disease was or what exactly happened in the 90s, but they came back out of it. They rebounded out of it. Around 2001, calves started happening again. And Stumpy, you could see, then bred the next year. The other thing that jumped out of this, look when the progesterone goes up. That's the beginning of the pregnancy. Look how far away that is from the yellow bar. Look at the timeline. These pregnancies are starting 6 months before we thought they should have. And that means these pregnancies are starting in a totally different month of the year than we thought. And that means the mating grounds are not where we thought, because these are migratory animals, right? If they're breeding in a different month of the year, they're breeding in a different location than where we thought, because they're in different places in different months of the year. So this was, again, just one animal. This was the very first baleen hormone data set. If you read the paper that I published about this, which was this very first paper in the lower left, I do not mention this oddity of the progesterone seeming to rise much earlier than we thought it should. I kept thinking I'd done something wrong, or I'd misinterpreted the data, or I just didn't trust it yet. We've now had a bunch of other studies on North Atlantic rights and on two related species, southern rights and bowheads. They all have much longer gestations than we thought they should have. So we're now in phase 2. I've kind of got my validations under control. I'm pretty sure I can measure the hormones. I'm pretty sure the hormones are telling me the truth. And we're now moving into what I call the descriptive studies phase of starting a new sample type, where you've got this new method, and you're just looking at the patterns that are there. You know, okay, now we can measure progesterones in baleen. What do progesterone patterns look like in a female, in a male, in a calf, in an injured animal? What do cortisol patterns look like in all those cases? So we have this phase of just sort of measuring these hormones and just describing the patterns that are there. This is a fun phase of research where it's sometimes thought of as sort of old-fashioned 19th century science, where you're just doing descriptive science and you don't have elaborate hypotheses about influences of the ecosystem on the animal yet. You're just measuring stuff and seeing what's there. But I find it a really rewarding type of science to do, especially when you're working in a field where there's no information yet. Almost everything you find is new, and you immediately start finding some unexpected things. So I'm making a little pitch here that descriptive science has a lot of value, and you have to start with descriptive science if you're in a new field. You have to start by seeing what patterns are there before you then start trying to explain those patterns. Just first see what's there, and that alone is really worth doing. I'm just going to run through a few things we learned from these early descriptive studies. We've seen cases where females just seem to not ovulate for many years in a row. There's other females that do something different. There's cases, and Nadine has a bunch of cases like this for both bowheads and North Atlantic rights. We think we're seeing a lot of pregnancy failure. Pregnancies start, and then 6 months later they stop again, and that's way too soon. That pregnancy can't have gone to term. You have cases where a whale seems to be doing 4 pregnancies in a row in 4 years in a row. This is not supposed to be possible. Either those calves are dying or the pregnancies are failing partway through. So what we're learning is whales start pregnancies way more often than we realize, and a lot of them fail, and we never knew that they even happened. You have to look at the baleen to see that those pregnancies happen. If you're just following the whale around, you'll never know. And then there's this longer gestation. I want to show you an example of this really long gestation. This paper just came out from a great grad student at the University of Pretoria in South Africa. And she's working on the South African population of southern right whales. I'm just going to draw your attention to the black line in that graph. And if you can see the little tiny font there with the years, these shaded bars here are pregnancies. See the progesterone's going up, that's a pregnancy starting, then the pregnancy ends. It goes up again, then it ends. These are 3 pregnancies in a row. Each of these 3 pregnancies is 22 months long, 22 months long, when we thought it was going to be 12 or 13 months long, so nearly twice what we thought. All the right whales and bowheads, all this time, we've all thought that they take a year off between calves. You often would hear it described as, oh, right whales and bowheads have a calf every 3 years. They're pregnant for 1 year, they nurse for 1 year, and then they take a year off. And then they start the next pregnancy. They've never taken a year off. They start the next pregnancy right away. It's a much longer pregnancy. They don't have a year off. They are pregnant for nearly 2 years, nurse for a year, have like 2 months off, immediately start the next pregnancy. The pregnancies are starting way earlier than we thought, and that means they actually don't get a year off at all. This also changes our estimates of where and when they're breeding, and that turns out to be important for population management. Now maybe the breeding grounds are in a different place than you thought. And also it's turning out to be important for population models. We now know they can never breed any faster than a calf every 3 years. That's the fastest they can go. If the pregnancy and the lactation add up to be 3 years, that's the fastest they can go. So that's important to know for estimates of, what's the fastest possible reproductive rate for a recovering population. So all of that from one animal, or 2 if you add in my North Atlantic right, all of that just from this, descriptive study. So descriptive science matters, and you can learn a lot doing descriptive science. I want to show you just a few other snippets of things you can learn just by looking at the patterns that are there. I want to spend just a moment on males, because males have stories too. These are 3 different males. We have a North Atlantic right on the left. The other 2 are bowheads. There's a young bowhead who's 18 years old. That's young for a bowhead. Bowhead males are supposed to hit sexual maturity at the age of approximately 20 years. This guy was 18 when he died. I'll come back to him in a second. And then on the far right was a really big old bowhead. Our best estimate is he was about 150 years old. So this is the longest lifespan mammal on the planet, the bowhead whale. They can live over 200 years old, and that guy we think was about 150 when he died. So this is testosterone data from these 3 males. And again, I was just describing what does testosterone look like in these animals? Again, just descriptive science, right? But right away you see the most prominent thing that leaps out is T-testo is going up and down, and it's going up and down regularly, and it turns out this exactly matches the cycle length of the isotopes. These are annual testosterone cycles in all 3 of these males. Males have testosterone that goes up during breeding season, and it comes down in the rest of the year. This should not have been a surprise, because that's what all seasonally breeding mammals do. But for some reason, this seemed to be a surprise to the whale community. So male whales have testosterone cycles. Testosterone's only high for a certain part of the year, and it's low at other times of the year. So that was cool to find out right there. But then, again, you just stumble across these other findings when you're doing descriptive studies in a new field. This guy, if you see the little tiny red arrows in the graph at the left, he was sick and he got entangled in a certain year, and his testo peak the next year was almost not there. He nearly skipped a breeding season. So we're seeing there a case of reproduction being disrupted by stress, and especially by stress the year before. This guy, okay, he was 18 when he died, and his death is at the very right side of his graph, the guy in the middle. The right side of that graph is the date he died. Bowhead males are not supposed to be sexually mature till about the age of 20. He's already doing T cycles. And if you count backwards, if you figure out the age he was, if you go backwards, 17 years old, 16, 50, he was T cycling when he was 6. And they are not supposed to be sexually mature then. And in most mammals, you don't start doing these annual T cycles until you're sexually mature. So this pattern was seen in all the young bowheads. They're all starting their testosterone cycles at less than half the age than we thought they would. I don't think they're making good sperm yet. I don't think they're socially competitive yet. I don't think they could actually breed, but it's really interesting to me that their testes are coming online and making testosterone and doing this annual cycle long before we thought they would ever be capable of that. The big guy on the right, his T cycles are crashing, and they're also getting more erratic at the end, and his baseline's coming up. There's a lot of weird things going on with him toward the end. He is a really, really old individual, And you see this sort of pattern sometimes in those species that have reproductive senescence, which means species in which the oldest individuals stop breeding. And that's not all mammals. In fact, it's a minority of mammals that have reproductive senescence, but it looks like it might be happening in bowhead males. And we have some other lines of evidence that indicate it's probably happening in the females as well. And then there's the timing. The T-peak tells us the timing of breeding. This guy's T-peak exactly lines up with the beginning of the long gestation of that female I showed you earlier. They're mating in summer, not winter like we thought. This guy turns out he gave us a clue about a subadult grounds where we think the teenage bowheads, males and females, they go to a certain bay in Arctic Canada, which is not where the big older adults go. It's like a teenage hangout area, and we think I don't know what they're doing. I don't know if they're practicing or what they're doing, but we're starting to think maybe that's a bay that should also be protected. And then this guy over here, the regularity of his T-peaks, again, helped clarify month of breeding and when and therefore where the bowheads are breeding, which is probably lower Davis Strait, late winter, early spring, we think is now where they're breeding. So all of that just from descriptive science. Let's see, I wanted to point out a couple other things before I close out for today. And I have one more little fun fact I'm going to throw at you for blue whales, but let me first point out, here's two graphs you've seen already. Here's that female southern right whale with her super long 22-month gestation. There's that big old bowhead male. I'm going to put calendar years on. 1980, 82, 84, look how beautiful those hormone data look. And that big old bowhead male, his first T cycle, whoops, I went forward and I meant to go back. His first T cycle was in 1987. And we didn't assay these plates till quite recently, so we were looking at 45-year-old hormone molecules here, and they were fine. So steroid hormones are really sturdy molecules. We knew that already. In fact, we store them at room temperature. In fact, when you buy pure steroid hormones from Sigma Aldrich Chemicals, the instructions say store dry at room temperature. And steroid hormones just last a very, very long time. As long as they're dry, keratin the interior of the keratin is a dry area. So this gave me some hope that we might be able to look much further back in time. And I don't have much more time to tell you about this other awesome project that Jen, where are you, Jen? Can you wave? There you are in the front row. Jennifer Jelinczyk, who has also just arrived here at MMI, she's doing a project on bowhead whales where she's got some baleen from the 1800s, and she has perfect looking hormone data from as far back as we think approximately 1856. It might be 1857, but 1850s. And we think that chances are good we're going to be able to get hormone data out of populations much older than that. So there's old baleen in the natural history museums, and there's old baleen in archaeological middens. And this is finally going to be the way we can find out what normal used to be. Remember problem number 3 of everything's different today? How do we know what's normal? We can look at populations of the past with baleen and with keratin samples in general. I'm going to give you one more example. I want to show you one bit of data from an animal that has a little bit shorter baleen. It's going to be blue and fin whales. And I'm going to skate over a lot of fun detail that I could tell you about this story. This is really a whole other talk, but I couldn't resist trying to shoehorn it into this talk. The Smithsonian has this amazing collection of World War II baleen that was collected in 1946 and 1947 by the Japanese right at the end of World War II. If any of you have forgotten your history, World War II ended in 1946. Remember that year? I'm going to come back to it in a second. And immediately, the whaling nations were right back in the Antarctic whaling again, and there was a lot of whales killed in late 46, 47, and 48. And the Smithsonian ended up with 1,600 baleen plates that the Japanese had collected. It was 2 plates each from 800 whales, 400 blue whales, 400 fin whales. And it's this extraordinary sample set that was lost and nearly thrown away twice and left to rot and was in this big jumbled heap. And finally, a great curator, a preparator at the Smithsonian took it upon himself to sort out this bewildering mess of baleen and get it all organized. It turned out it was all still labeled and the logbooks were all still there, and it's now available for research. So there I am on the left. The person way at the end is me with this selection of baleen. There's 2 of my grad students sorting out some blue whale baleen and some fin whale baleen. We had baleen plates from, in the end, there's 1,600 baleen plates there. We have data from 18 that I'll show you because it takes a long time to do each plate. So just a little tiny piece of the collection. I just want to jump to my corticosterone data. So we were originally trying to, we had all these grand hypotheses along the lines of maybe they get more stressed when prey changes, when sea surface temperature changes, when the ecology changes, when the oceanographic conditions change. And so we sampled all the way down these plates. You can actually see the sampling grooves in that blue whale plate at the top. We had samples every centimeter from these plates. And we ran a bunch of hormones. We got really interesting progesterone and testosterone data. But I'm going to show you the corticosterone data. So this is the first stress hormone data I've shown you. We could not find any signature of cort changing with any ecological measure that we could think of. Prey, sea surface temperature, whether or not there was an El Nino or a La Nina. Nothing seemed to influence the cort. But when we just threw year into our model, we got this strongly significant effect that cort has a year effect. Corticosterone changes with calendar year. And here I'm showing you just a big scatter plot of our data from our first 10 animals. And I think you can see cort is higher in the last couple years. The scatter goes higher up. There's still a lot of variation, but the scatter is going higher up in the last few years. Let me just show you two individuals. Here is a typical male. And this is his cort by year. And I think you can see it's, there's statistically no difference in his cort level from 42 to 45, and then it ticks upward in 1946. Remember that year? And it stays high in 47, 48, and now a female. This was actually the more common pattern where it was a little high in 42, then it was low in 43, 44, 45, and then it ticks upward again in 1946. Tell me again, what happened in 1946 in the world? Whaling started again. World War II had just ended. So there was no whaling in the middle of World War II because all the whale ships were converted to the war effort. There was a little bit of whaling in 39 and 40, a little tiny bit in 41, and then no whaling, as far as we can tell no whaling at all in the Southern Ocean in 42, 43, 44, 45, and then whaling started again in 46. And the baleen plates we had were grown during World War II. They were collected in 46, 47, and 48, but they were grown during World War II. And we actually are now working with a team at the University of Hawaii that has some really big blue whale plates from that same collection, and they've got 37, 38, 39 in their data set. They have some of the earlier years, and they've been able to confirm what we also had found. If we divide our data just by was there whaling that year or not, that is the most significant effect we have on corticosterone was there whaling in the Southern Ocean, yes or no, in the year that this baleen plate was collected. This really surprised me, and I still don't fully trust it. I trust it more now that I know my University of Hawaii colleagues have found the exact same pattern with a whole different set of baleen, but for the same years. And we're actually going to be combining our data sets and publishing them together. So did whales find peace in war? I think the answer is going to turn out to be yes. And this, to me, was kind of an eye-opening example of a human impact can be stronger than any of the ecological impacts. And it looks like that's one of the biggest effects on the stress physiology of these animals was just, were they getting periodically chased by whaling ships or not? There's a lot more to it than that. I'm still hesitant about this result, but this is the patterns that we're seeing so far in the data. So I've been working on baleen hormones since 2014. That's when I first got that little scrap of information from those very first bowheads. Where are we now? So here's the baleen hormone literature today. We've now got 20 manuscripts out, and not all by me. There's a bunch of other teams also now using this technique. I know of another 8 papers that have either been submitted or about to be submitted, and there's at least 10 more that are being written, so the field is about to double in size. And I think it's really interesting to look back and see, first off, that it took 12 years. It takes a while to launch a new field. And look back at how it played out and the stages it's gone through. So you have to start with your validations. And I have here the summary of all sorts of things we've verified with validations. But we're not done with validations, and we're never going to be done with validations. Validations will take up the entire rest of my life, but we've ticked off some of them. And then there was the descriptive studies, and we're still, these phases actually all kind of happen simultaneously. You end up publishing data that's part validations and part description and part hypothesis-based science. But the descriptive studies, in short, have been really interesting. We've found some dramatic things about reproductive biology in these animals that we could never have found with just like one respiratory vapor sample or 2 poop samples. This repeated samples over time that baleen gives you is so valuable. We're now finally moving into what I call phase 3, where we're looking at what's causing the patterns that we see. We're moving beyond just describing the patterns and asking, okay, why do the patterns look like that? So you just saw that with cort lines up with whaling, right? That was an example of this phase 3. And there's a lot more studies now either underway or being planned that are looking at the effects of ecological change on stress and reproduction, why different individuals have different patterns, what's driving those individual differences, what's driving the calendar year effects. Phase 3 is getting complicated. You need a lot more than 1 or 2 whales. You need a lot more different things that you're measuring in the baleen, so more analytes. You end up needing a lot more sophisticated statistics. You've got many individuals, all with repeated samples over time. Each one is covering a different set of years. It gets complicated. So you need to build like pretty elaborate models to tease out all the influences. But it's starting to happen, and I'm really excited about this phase 3, which is largely not going to be me. It's going to be other people that hopefully I get to collaborate with that are doing a lot of these studies. But I've got a couple irons in the fire for these phase 3 studies as well. So it's been really interesting and exciting to be able to be part of launching a new field. And we're still not done. There's new biomarkers I'm trying to develop. Like I said, I'll never be done with validations. I feel like we need to all be teaming up more. Baleen generates a ton of samples. It takes a lot of work. I would like to help someone else plan a baleen hormone research consortium where we can all pool our data together. I want it to happen. I don't have the time to do it, but I think it could be really, really useful. And really, we've all been collaborating sort of informally up till now anyway. It takes a village to launch a field, and it takes more than a decade to launch a field. But in the end, it's really worth it. If I could give this one message to the young scientists who I know are watching, it's that it really pays off to put the legwork in to get a new technique rolling and to do those validations and do those descriptive studies. They turn out to be incredibly valuable, and all those things are publishable. People will tell you, don't publish methods papers. No, publish methods papers. Publish them a lot. Publish them a ton. They actually get cited a lot, and then you get to see the whole field mushroom, and it's really rewarding to be part of a project like this. All these studies came about because whales died, and I have to acknowledge that. It's always a tragedy when a whale dies, but I do feel it is our duty to learn everything we can from every such case so that hopefully we can help them not die in the future. I'll stop there and take any questions. Thanks very much for listening.
That was amazing. Thank you so very much. I know we're up against time, so if anybody needs to sneak out, you can head out the back of the auditorium. And for folks online, we're going to start with your questions because everybody in the room can kind of come up after. So Roseanne, it looks like you got a question coming in our way.
Yeah, great work, Kathleen. Thanks for the shout out to descriptive science. I know this talk was mostly about marine mammals, but I was wondering if you could share some core research questions you had with elephants.
Oh yeah, elephants. So I started working on elephants. We were looking at fecal hormones originally as a way to be able to get samples from wild elephants without having to tranquilize them. And I got to be part of actually a really interesting group of scientists who specialize on elephants. One of our we had 2 major things we were working on originally. One was the effect of stress in the wet season versus the dry season, so ecological influences on stress physiology and reproduction. But we also stumbled across a really fascinating and rather disturbing case of lingering long-term stress from poaching that had happened years ago, but the elephant social systems were still disrupted because all the matriarchs were gone. And you had all these young elephants with no matriarchs, and they still weren't breeding yet, well, more than a decade after the poaching had ended. So we were able to get at that through stress hormones and reproductive hormones in their poop. I'm now actually working, even though I'm now here at the Marine Mammal Institute specializing on marine mammals, I just moved from the East Coast where I was working with another elephant team. We have gotten good hormone measurements out of elephant tail hairs. That's why I had a picture of an elephant tail in one of those earlier slides. I've been working on elephant tail hairs and also their toenails. Turns out you can get hormones out of toenail clippings from elephants. And we were able to look at stress of a long-term disease that one elephant had and how she'd recovered from that disease and her stress hormones went down and how that had affected her reproduction. The other thing that I did a lot of work on in elephants was trying to figure out the variation in the estrus cycle length in different females, because it turns out there's a lot of individual variation in how long their estrus cycles are. They're really interesting animals. I keep thinking about elephants while I'm working with whales because they have very long lifespans and very long reproductive cycles and very slow calf output. So they've actually got some similarities in sort of the slowness of their reproduction. So I find elephants and whales similar in some ways. I still think of whales sort of as large swimming elephants.
That's We're going to take one more question online, and we'll get one in the room, and then we'll wrap up. Go ahead, Roseanne.
Do you have to do isotopes every time you do hormones to validate the timing?
I don't have to, but I prefer to. So anytime I am able to do isotopes, I like to look at the isotopes of each individual whale, because there is some individual variation in the isotope cycles, and I still am, a lingering concern for me is the baleen growth very consistent, or can you have a year when your baleen's growing unusually slowly, or a year when it's growing unusually fast? And it helps reassure me if I have the isotopes for that individual whale. There is a little variation. It looks like it's not too huge. But there have been cases where we have basically someone else has done some isotope work on that species, and we've ended up concluding like, okay, in adults, baleen growth rate is about 16 centimeters a year for females of this age. And sometimes I just use that average growth rate, and I can develop a pretty good timeline from that, at least enough to see where the years are. I'd be less confident about being sure I could separate like winter from spring if I'm using a rough average like that. But I can at least get a pretty good guess at how many years are on the plate with those species averages.
Nice. Questions in the room? Want to be the one question? Okay. And while we're getting to this, do you have a slide with your contact information on it?
Oh, I didn't think to put it up there.
Okay. So for folks online, if you have questions that we're not going to get to, you can actually go to the Marine Mammal Institute and pull up Kathleen's email.
Yeah, and any second now, I will populate my page with more information. So I just landed here, and there's still not much information about my lab online, but that will be appearing soon. And please do feel free to reach out and contact me. Also, I have to apologize. The reason that we're running out of time for questions is that I wanted to show you the blue whale court data from World War II. I knew I would run out of time if I squeezed that in, but I couldn't resist. I hope it was worth it.
I get how you were.
Test.
I get how you were measuring the keratin, and most of the graphs were per weight on grams. So I'm trying to figure out, and I apologize if I missed it, how you're able to calculate expiration and poop. Is it per volume? And where do you try to get that as, or does that matter? It seems to me like it would make a difference.
There's a category of samples that I call the hard sample types, which are solids rather than liquids. So like urine is a liquid, blood is a liquid. When I'm measuring hormones in those sample types, I measure it per unit volume of the sample, like per milliliter of blood or per milliliter of urine. But for poop and for baleen and for hair, and there's a couple other sample types like that too, I end up drying the sample. So for poop, we freeze dry it all. We get all the water out, and then we have a dry weight. And then we weigh out a certain number of milligrams of that dry fecal powder. And in the end, we express our hormone concentrations as nanograms of hormone per gram of dry fecal powder. With baleen, it's already a very dry sample type. We do actually, if it's small enough to fit it in the freeze dryer, we freeze dry it too. But it's basically already dry. We just we use the Dremel to pulverize a little bit of powder, and then we weigh the powder. What we actually put into the extraction process is we weigh out 20 milligrams of the powder exactly. And then that's what we extract the hormones out of, and then we measure the hormones that were in that 20 milligrams. But then we do a conversion so that our final data is expressed as nanograms of hormone per gram of the baleen powder. So it's per gram of dry sample in the end.
All right, I think we could ask you questions forever, and I'm so glad that you're here so that we can have you come and speak to us again. But one more round of applause for Kathleen, please.
Thank you, everybody. Thank you.
All right, come next week and learn about lava. Awesome. Thank you very much, Kathleen.
Thanks, everybody. So anyone who wants, please come up and take